Okay, in this video we are going to pick up with fever. Okay, so there we go. All right. So fever is body temperature above normal. Okay, typically normal body temperature is between 97 and 99 Fahrenheit. Anybody that's got a fever is called a febrile. This is a critical warning sign of inflammatory responses that are happening somewhere in the body. It is an innate response, so there are chemicals called pyrogens that are released from the damaged cells or even certain bacteria. Those pyrogens act on the hypothalamus and they reset the hypothalamus to a higher range. All right, so this is what's going to kind of put us into positive feedback. After that onset of pyrogens, the hypothalamus is going to interpret normal body temperatures being too low, and then we're going to trigger a negative feedback, which is going to, basically start the fever. Now, sometimes this is why people get that chills when the fever occurs, because their normal body temperature is, the brain is reading that as too low. So that's kind of where that comes in. The hypothalamus is going to trigger responses that elevate body temperature to a higher range. Some familiar signs would be shivering. Increased muscle activity, of course, is going to generate heat and bring that body up to a new set point. Now, eventually the fever will break, and that's when the hypothalamus has been reset to normal. It causes the hypothalamus to realize that the febrile temperature is too high, so it triggers negative feedback to lower that temperature. Mechanisms can be sweating, dilating in the blood vessels, which makes the skin sometimes appear red or flushed. Okay, and again, most of you have experienced this with fever. Okay, now we're going to move into cell-mediated immunity. So we're going to switch from innate to adaptive. Now remember, adaptive immunity has two different arms. It has the cell-mediated arm and it has the antibody-mediated arm. So we're going to start with cell-mediated. Now, there's different classes of T cells. You're going to have helper T cells and you're going to have cytotoxic T cells. Okay. Now, these are primarily responsible for responding to cells that are infected with intracellular pathogens, like viruses or bacteria that are in the cell. They also respond to cancer cells and any foreign cells like those that would come from a transplanted organ. So that is who cell-mediated, these type of cells are actually going to respond primarily to. Cancer, foreign cells, and intracellular pathogens. Now, T cells we know are made in the bone marrow. They leave the bone marrow and go to the thymus to mature. Now, each population of T cells that can respond to an antigen is called a clone. There are millions of different clones in the immune system, but only a few cells of each clone exist in the body at a given time. Some T cell clones can recognize and respond to pathogens, and then others cannot. What the thymus does is it screens the cells, and it helps mediate the destruction of clones that cannot recognize antigens. So if these T cell clones cannot recognize antigens, they're destroyed. Okay, the thymus is responsible for screening. It basically gives them a test. All right, and by doing this, the thymus is making sure that an individual is what we call immunocompetent, and that means it's able to mount a normal response to foreign antigens. We never want our cells to mount responses to self-antigens because that would put us in an autoimmune problem. All right, so thymus is going to make sure we're immunocompetent, which means our T cells can respond to antigens. Now, the mature naive T cells, naive means they've not yet encountered an antigen, are going to migrate, they're going to leave the thymus, and they're going to go to a lymphatic organ, and they're going to wait to come in contact with a foreign antigen. Okay, so right here kind of shows you, this is in the bone marrow, cells of that lymphoid line divide in the bone marrow, producing lots of T cells, immature T cells, and they're going to migrate to the thymus, and this is where they become mature. The thymus is going to stimulate that maturation, and as well as destroying any self-reactive T cells. Now, that test I was telling you about over here, right here, when the thymus screens those cells, any T cell that responds to a self-antigen is destroyed. All right, we do not want self-reactive T cells. That means our T cells would attack our own cells, and that would not be good. Okay, once they pass the screening from the thymus, they go back into the bloodstream and they migrate to a lymph node, and that's where they wait to come in contact with an antigen. Okay, notice their blood, right? Blood's our transport. It takes it from the bone marrow to the thymus, takes us from the thymus to the lymph node. Okay, all right, so we know how they mature. Now let's talk about the response. All right, only certain antigens, which we call immunogens, are capable of generating a response from this immune system. Self-antigens are antigens that are present on your own cells. They are not immunogens in your body. All right, now in somebody else's body, they would be immunogens, but in your own body, self-antigens are not. Those are your own antigens that are present on your cells, so that's what we call self. Now, haptens are really small antigens that are immunogenic only if they're attached to a protein carrier. All right, so this is kind of where it gets into different types of antigens. Now, on every T cell surface, you have a receptor, a T cell receptor. That receptor binds to the specific antigen before the cell can become activated. Okay, now there's a unique portion of the antigen to which the receptor binds. It's called the antigenic determinant. Okay, so the antigenic determinant is actually the part of the antigen that binds to the T cell receptor. Okay, and we're going to look at some pictures in a little while that'll kind of help with that. Now, T cells only interact with pieces of the antigen that are bound to the MHC molecules. Okay, they are bound to glycoproteins, which we call major histocompatibility complex, or MHC molecules. All right. MHC molecules are kind of like docking sites. And remember this term, glycoprotein? Glycoprotein means you got a protein with a sugar chain attached, or carbohydrate chain attached. That carbohydrate chain is a unique chemical ID for each cell. So when it's connected to a protein, it becomes a major histocompatibility complex. They're found on every nucleated cell in the body except erythrocytes. Red blood cells do not have MHC molecules. Red blood cells just have their antigens. That makes blood products a lot easier to donate than organs and tissues, because with blood, you just have to make sure you have the right antigen, A, B, or O. All right. With organs and tissues, you have the blood type, you have the antigen, and you have the MHC molecule. And the MHC molecule is a docking site. Okay, and it takes specific parts of the antigen and it docks them on that site, and then it displays them to the T cells. Now, back up a second. Okay, why are we taking the antigen, docking it on an MHC molecule for a T cell? Well, T cells are the only cells in our bodies that can determine whether that antigen is self or foreign. Nobody else can do that. So these MHC molecules have to take the antigen, digest it, and then dock it on their site. Okay, and these are all macrophages. Dock it on that MHC molecule site, and then the T cell comes by and can decide whether that's a self-antigen or if it's a foreign antigen. If it's a self-antigen, nothing's going to happen. We don't react to self-antigens. If it's a foreign antigen, we do have it happening. Okay? So that's kind of how that works with the T cells. All right, let's keep going here. Okay, there are two types of MHC molecules, class 1 and class 2. All right, now class 1 MHCs are found on the surface of the plasma membrane on nearly all nucleated cells. Okay, now they are going to present what we call endogenous antigens. Endogenous antigens are those that are made inside the cell. So it could either be a foreign antigen that was present on a pathogen that lives inside your cell, like a bacteria, or a foreign or a self-antigen that was encoded by your DNA. It includes normal self-antigens, foreign cancer antigens that are made by mutated DNA, and then of course viral antigens. Okay, the bottom line is any antigen that is inside your cell, they are going to be presented by the class 1 MHC molecules. Now, generally, cytotoxic T cells only interact with class 1 MHC molecules. So, here's the deal. Right here, I love this picture. This is the diseased body cell. This purple doohickey here, that is the class 1 molecule. The pink thing, that is the endogenous antigen. Endogenous means it has to come from inside your cell. All right, right here is the T cell. It is a cytotoxic T cell because that is the one that interacts with class 1. Okay, right here, this kind of aqualish green, that is the T cell receptor. So what happens, this disease body cell has this class 1 docking site. It's going to dock this antigen on it, and then the cytotoxic T cell is going to come and connect. The receptor connects with that antigen. This T cell is going to decide whether or not that antigen is self or foreign. If it is self, nothing happens. We don't attack our own antigens. If it's foreign, then the body's going to get ready to launch an attack. Okay? Now, the other type of MHC is the class 2, and they are found only on the surfaces of antigen-presenting cells. Remember, APCs. These are basically macrophages, dendritic cells, natural killer cells. All right? Now, class 2 MHC molecules are going to present exogenous antigens, or those that the cells take in by phagocytosis. Exogenous antigens are nearly always foreign because it's ones that the macrophages or the APCs, right here, the APCs, they've ingested them. They've taken these cells in by phagocytosis. So these antigens did not come from inside the cell. So they're almost always foreign. Okay. Now, helper T cells generally only interact with class 2s. All right. So here we go. Here's our picture. You have the APC. All right, this is that antigen-presenting cell. Here's the class 2 MHC molecule. Here's the exogenous antigen, which means it came from another cell, not one of our cells. And then here's your helper T cell, and then of course there's the helper T cell receptor. Okay, this helper T cell is going to bind and read this antigen. It's almost always going to read it as foreign, and so this is going to launch an attack. All right, so class 1 MHC molecules and class 2, very, very, very important that you know the difference. Okay, now how do we respond? Once we know we've got a foreign antigen, what is the T cell response? Well, this is activation. This is what happens. The cells display the antigen on their MHC molecule. So again, here's your T cell. All right, here's your body cell. This is, again, typically in the lymph node. Here's the T cell receptor. This is the MHC molecule. Of course, the pink thing's the antigen. So the cell displays it on their MHC molecule. The antigen binding site binds to that receptor, either for a helper or a cytotoxic T cell, depending on what type of MHC molecule it was. And then you have this thing called a co-stimulator. See this deal right here? This is a co-stimulator. The helper or the cytotoxic T cell binds to the co-stimulator, and then it becomes activated. Okay? Then the activated T cell clone proliferates and differentiates into either effector cells or memory T cells. Okay, so once this co-stimulation happens, they're activated. And so now our T cells are going to start dividing. Okay, and they're going to divide and they're going to produce clones. Okay, some of the clones become effector T cells. Some of the clones are going to become memory T cells. All right, now the memory T cells are not going to be involved in this attack. All right, they're going to respond quickly and efficiently to another exposure to the antigen. They do not need a co-stimulant. That's the beauty about the memory T cells. They don't have to go through this step. They just automatically can start taking care of business once you have a repeat exposure. Now, these effector T cells, they're the ones that are going to be, now they're going to go and attack these antigens. Okay, so this is how we're attacking. Now, proliferation just means to divide, to make more. Differentiation means we're going to differentiate into either an effector T cell or a memory T cell. So differentiation means we're going to turn into a specific type of T cell. All right, so that's what that means. Now, what happens with helper T cells? Because that was kind of general. All T cells. Let's talk about what happens with the helper T cells. The helper T cells and our cytotoxic have very different roles. Now, they do interact and help each other, but they do have different roles. The helper T cells have no phagocytic ability. They have no cytotoxic ability. They basically exert their effects by secreting cytokines that activate and enhance the other components of the immune system. This is how they help the immune system. Okay, now helper T cells are required for normal function of all components of the immune system. So this is what the helper T cell does, is it secretes massive amounts of cytokines. See all these little bubbles? These are all chemicals. Massive amounts of cytokines like interleukins. Okay, remember the interleukins? They're going to stimulate macrophages to enhance phagocytosis. They also are going to stimulate macrophages to release more interleukins with that to help with phagocytosis. They're going to activate proliferation of cytotoxic T cells. They're also going to stimulate B cell proliferation, which is getting into the adaptive immunity, which is, we're not exactly there yet. But helper T cells act in both innate immunity and adaptive immunity. All right, so it's really important to point out there. Now, cytotoxic T cells is very different. Their primary function is to kill other cells, okay? Specifically those that have a foreign antigen bound to their class 1 molecule. So they can detect any abnormality in the cells, which is really important for detecting cancer, foreign cells, and any cell that's infected with a virus or bacteria. So basically, you have your normal cell here, because that's who has the class 1, MHC class 1 molecules. The cytotoxic T cell binds to the target cell. Right here, oops, sorry. Let me go back. It's going to read this, okay? So this is reading this antigen. The T cell then releases a perforin. All right, here's all these little perforins, and that's going to form a pore, which is basically a hole in the plasma membrane. Then the cytotoxic T cell is going to blast it with all these little enzymes, these little triangles. They're going to blast these enzymes through the pore, and they're going to get into the DNA of the target cell. And then basically it causes the target cell to degrade, basically just disintegrates. Okay, well, once it starts disintegrating, the T cell detaches and is going to go look for somebody else to destroy. Okay, the cytotoxic T cell is going to detach and go search for something else, while this cell is now basically destroyed. Okay, now macrophage is going to come along and get rid of the rest of this, whatever's left. All right, so very different, the cytotoxic T cell and the helper cell, very, very different. Okay, all right, we're going to stop there. We'll get into antibody-mediated in the next video.